Hey, hold up. Yeah, no. Squad. Gang. Yeah. Hold up. Na. Yeah, na. Hold up. Is this biology or chemistry? High off life or...
All right, welcome back, people. Let's not waste any time. So here are the things that we will be covering in this video. First, we're going to look at an animated tour of the cell. Then we'll watch a second clip that shows some of the special organelles that are dedicated to plant cells only. We'll compare the plant and animal cell organelles. And then finally, I'm going to cover something called the endomembrane system, which includes several organelles. So we'll cover the nucleus, ribosomes, the endoplasmic reticulum, both the smooth and the rough, the Golgi apparatus or Golgi bodies, and finally lysosomes and vacuoles.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells. Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
So our animated friend said a lot, and she went over a bunch of different organelles and their functions. Don't freak out. That was supposed to just show you an overview of how everything is working internally. So now we're going to shift our focus to each of those things that she was talking about. But again, in this video, we're going to stop with the endomembrane system. So let's focus our attention right now on the slide that's in front of us, figure 4.7-1. This is us exploring a model eukaryotic cell. Specifically, it's an animal cell that we're looking at. So when we look at this picture, we have to remember that this is a model that is all inclusive. So by that, I mean all of the possible organelles that can be present in an animal cell are. Not every animal cell has every single one of these organelles present. Also, the number of each organelle can fluctuate depending on the job of the cell. So for example, if we were to look at a muscle cell, we would encounter a large number of mitochondria and we would not see any flagella. So the question is, why or how do you know that? Well, because muscle cells are associated with doing work, and that work requires a great deal of energy. So you think about something like exercising or even just walking. The ability of a cell to take in oxygen and glucose and then turn them into being able to break apart ATP molecules for energy release is crucial to that cell's particular function. Muscle cells are not swimming around, so they don't need the flagella whip-like tail to make that happen. Take a second, just look at all of the different organelles that are listed, and you'll see that there's a bunch, but you'll remember them based on their job, what they do, and how important each one of them are. Figure 4.7-2 is exploring a eukaryotic cell once again, but this time we're looking at a plant cell cutaway. Remember that this is just a model, so every possible organelle that a plant cell could contain is listed here on this slide. We have to stop for a second and think about plant anatomy, and we'll realize that not all of these organelles are necessary for every part of the plant. So one specific part of the plant that is underground is the root. So plant roots, they are not in contact with the sun. They don't need the exposure of the sun. So if we think about the anatomy of roots and we think about the fact that they're underground, we realize that root cells don't need to undergo photosynthesis. Therefore, they wouldn't need chloroplast. They don't need any organelle present that's going to essentially take the sun's energy and some carbon dioxide and turn them into carbohydrates for the cell to be able to break down later. Now that we have noted some of the differences in plant and animal cells by looking at the two models, we're going to shift our focus into things that they both have in common. So in other words, we're going to study which organelles that they both have present. We start with the nucleus, and it's appropriate to start here because you should recall from the last video that this is the first organelle that we look to be able to break cells into two domains. So this is what you need to know about this part of a eukaryote, the nucleus. The nucleus contains most of the DNA in a eukaryotic cell. Ribosomes, which is another like organelle that we'll talk about, they use the information from the DNA to make proteins. The nucleus contains most of the cell's genes, and it is usually most noticeable when you're looking at a cell under a microscope. So the nuclear envelope is something that encloses the entire nucleus. It separates it from the cytoplasm. The nuclear membrane is a double membrane. So each membrane consists of a lipid bilayer, just like the phospholipid bilayer. There are nuclear pores that regulate the entry and the exit of molecules. The shape of the nucleus is maintained by the nuclear lamina, which is made up of protein. So this is what we see. We see each of the parts that I just called out. First of all, you can see the nuclear envelope. There's an inner and outer membrane. They also are showing you here nuclear pores where things can enter and exit fairly easily. They give you a close-up of a nuclear pore down here in this box. And then we're also seeing chromatin, which is just wound up DNA. Let's clarify some vocabulary at this point. We're going to look at what is the difference between chromosomes versus chromatin versus just straight up DNA. So chromosomes, they are nothing more than a condensed version of DNA. What you need to know about humans is that we have 23 pairs of chromosomes, and each one is a long string of DNA. Chromatin, though, is what we call the DNA and the protein together. DNA is wrapped around these proteins called histones, and that is what you see when you look at the DNA being tightly wound around this like weird looking structure. Another word on here that we have to pay attention to because it's bolded is the nucleolus. So what is the nucleolus? This is where we make rRNA, and that extra R in the front that I'm saying stands for ribosomal. So we're making ribosomal RNA in the nucleolus, and those are the instructions that we need to make new ribosomes. So in addition to all of the rest of our genetic material being found in the nucleus, we also have instructions to make new ribosomes in the nucleolus of the nucleus. We ended the last slide talking about ribosomes and ribosomes being made. Figure 4.9-1 is dedicated just to the ribosomes. So ribosomes are protein factories what they do is they unite. There's two parts to it. You have the large subunit and the small subunit. When the two of them are together, then we have a full ribosome. So they're complexes of ribosomal RNA and protein, and ribosomes carry out protein synthesis in two different locations within the cytosol, or within the cytoplasm, excuse me. One of those locations is the cytosol. So when we find a ribosome in the cytosol unbound to anything else, we call that a free ribosome. There are pictures of them within this transmission electron microscope picture here. So you notice that the free ribosomes are located like all throughout. They're all of these little black dots that are not connected to this like extremely dark like double black line that you see. So those are the free ribosomes. Now, the other ribosomes that we talk about, they're on the outside of the endoplasmic reticulum, which is right outside of the nuclear envelope. And these ribosomes, they're not free, they're bound. So they are actually stuck to the ER, and they give rise to a type of endoplasmic reticulum, the rough endoplasmic reticulum. So we call it rough ER because the ribosomes give it this appearance of being bumpy. Once we figured out what the ribosome mostly is made out of, and the fact that there are two units, a large subunit and a small subunit, we're able to enter that information into a system, and a computer model will give us an idea of what the ribosome looks like chemically. So figure 4.9-2 is just a computer model of a ribosome. We've looked at plant versus animal cells, and we have also discussed the nucleus and its structure. Now we're going to move on to concept 4.4, which covers the endomembrane system. So the prefix endo literally just means inside. The endomembrane system is nothing more than a system of membranes connected within the cell. Collectively, the job of the endomembrane system is to help with the making and movement of proteins within a cell, but it also aids in metabolic functions. When you hear the term metabolic function, you should automatically associate that with breaking molecules apart. That's an act called catabolism, or putting molecules back together again, which is called anabolism. Individually, all of the parts that I'm going to talk about have their own purpose, but it's important to remember the bigger picture while we're looking at the smaller working parts. The endomembrane system includes the following organelles. So we have the nuclear envelope, which is just a part of the nucleus. We've already looked at its structure, but it's still involved here. The endoplasmic reticulum, we'll talk about smooth versus rough ER. The Golgi apparatus, the lysosome, vacuoles, and last but not least, the plasma membrane. Some of these parts are so close together that we consider them continuous. The parts that are not so close together, we use something called vesicles to carry molecules back and forth as needed. So the vesicles are just little packages that pick up things and drop off things. The title of this slide is befitting. The ER, or endoplasmic reticulum, is a biosynthetic factory. Let's do a word breakdown of biosynthetic. So bio meaning life, and then the synthesis part of it means you're making something. So the endoplasmic reticulum is involved in making parts that give rise to a cell, and the cell is the basic unit of life. The nuclear membrane is in direct contact, or it touches the endoplasmic reticulum. If you could walk around in a cell and start from the nucleus, you would walk out one of the nuclear pores and you would walk directly onto or into the endoplasmic reticulum. So the first bullet on this slide gives us a factoid that helps us picture how much of the cell is dedicated to this particular organelle. If we could measure all of the membranes that constitute the cell and organelles within the cell, we would find that the membrane of the ER makes up more than half of that number. We separate the ER into two categories based on the appearance of the two different regions. We also consider the job that each carries out. Let's take a look at location, and then let's point out physical appearance of both in the next slide. So if we take a look at the overall model cell here, you'll notice that the part that is being emphasized is the ER, and they color it in blue. Everything else is whited out. So here they're showing you where the nuclear envelope is in relation to the smooth ER and the rough ER, and they're in direct contact with the nuclear envelope. There are various parts of the ER. They each have a name. Really what's important though is to understand that the ones, or the piece of the ER that's studded with ribosomes is called the rough ER, and again the smooth ER lacks ribosomes, therefore they call it the smooth ER. They show you what a transport vesicle looks like. It literally is just a piece of the ER's membrane that pinches off and will send whatever is made to the next organelle. The picture on the right is again a transmission electron microscope image of what the ER looks like. They point out what is smooth and they point out what is rough, and you're able to see the ribosomes on the rough ER quite clearly. Smooth ER, you see that it lacks those same ribosomes on its surface. Now that we have an idea of the location of the endoplasmic reticulum, and we also have an idea of what the smooth ER looks like versus the rough ER, let's talk about the function of both. So the function of the smooth ER is as follows. It makes lipids. It is able to break down carbohydrates. It also is capable of detoxifying drugs and other poisons that are foreign to your system. And last but not least, it is responsible for storing calcium ions. The rough endoplasmic reticulum, on the other hand, has these bound ribosomes, and they push out something called glycoproteins. The glyco part is referring to the carbohydrate portion, and then protein is talking about protein. So protein covalently bonded to carbohydrates, that's what a glycoprotein is. Transport vesicles are also listed here. So first, I guess I should have highlighted that, and then transport vesicles. So the transport vesicles are responsible for taking the proteins that may be made and pushing them out into the surrounding membranes that need to further process, package, or use the proteins that have been constructed. So in that way, the rough ER is very much a membrane factory for a cell. If we were to continue on our walking tour, the next organelle that we would encounter as we walk through or across the rough ER and the smooth ER would be the Golgi. So there are a few names for this organelle, and you will hear them being used interchangeably. You may hear it being referred to as the Golgi apparatus, like it is in this slide, the Golgi body, or the Golgi complex, or just simply Golgi. And the last one is the one that I use the most because it's less it's less cumbersome than trying to say some of the others. So the job of this organelle is threefold. It can modify products that are coming from the endoplasmic reticulum. It also helps in the manufacturing of various macromolecules, so processing or helping to contribute to the making of proteins, carbohydrates, lipids. That's what it does. Last thing that the Golgi is capable of is sorting and packaging different products into transport vesicles, which would be moved to the next organelle for further processing. So now that we've listed the functions and described where it is in relation to the endoplasmic reticulum, we should look at the organelle up close. Figure 4.11 does the same thing that we did in looking at the model of the ER. So you see the model cell again here. And then you'll notice that now it's not the ER that's being colored, it's the Golgi that is being covered, because that's the part that we're interested in right now. So they show you where it's located in relation to everything else, and then they blow up the image so that you can see very closely kind of what it would look like. So there are two different sides of the Golgi. You have the cis side, which is the place where things from the ER would be received. Notice that they're in these small bubbles. Those are those transport vesicles that are being alluded to. You'll also notice that there's a trans side, and the trans side is where things are going to be pushed out after they've been packaged and made, or made and packaged. Again, to the right, we have another transition electron microscope photo of what this particular organelle looks like up close. Moving right along, we encounter a group of organelles called lysosomes. So lysosomes are made by the Golgi. Lyso has Greek origins. It means dissolving or loosening. And this is what the organelle does. It breaks apart molecules. So the organelle is made up of a membrane, and inside of that membrane we find proteins, specifically hydrolytic enzymes. The hydrolytic enzymes rip apart macromolecules. The enzymes within the lysosome have an acidic pH, but the organelle itself is not consumed by the enzyme when it's not doing the job of breaking apart macromolecules or old cell parts, because the enzyme is inactive during that time. So imagine, if you will, for a second, that there are a few molecules that are outside of the cell that need to be brought into the cell, and they need to come in so that the cell can, reorder them, repurpose them, basically break them down and then reconfigure them. So what happens is the molecules would sit like outside of the cell, right outside of the cell, and then the plasma membrane would engulf the molecules. It would kind of like, they call it phagocytosis. It means like the cell is eating. And the word is here, by the way, phagocytosis, bam, right? So what happens is the cell membrane will engulf the molecules by phagocytosis, and then the vesicle that forms would kind of like pinch off, and then it would be inside the cell. So we're not talking about molecules that are going in and kind of just like, free floating. We're talking about molecules that are actually enclosed within a piece of the plasma membrane. And so what happens is, as this vesicle, that's what a vesicle is, as this vesicle kind of moves through the cytosol, it would eventually encounter a lysosome, or it would move towards a lysosome. Once those two parts encounter each other, so once the vesicle and the lysosome fuse together, the enzymes inside of the lysosome become activated, and the molecules that were pulled into the cell are now able to be pulled apart and reconfigured, later in some other organelle. So that's the lysosome's job, just to digest whatever it needs to digest, or to break down whatever it needs to break down. So there's one cell type that I want to point out, and this kind of goes back to what we were saying about like the number of organelles and which organelles would be present in one cell versus another. I gave you mitochondria and muscle cells as an example. So here's an example for lysosomes. Think about your white blood cells. In case you did not know, your white blood cells are a part of your immune system, and their job is to capture pathogens like foreign invaders that are not supposed to be there. They engulf those invading cells, and they have a special organelle inside, what do you know, that is responsible for secreting, or rather meeting up with this pathogen and then activating its hydrolytic enzyme and effectively ripping the pathogen into pieces. So there are a lot of lysosomes in white blood cells, and that is how a white blood cell is able to do its job. So it's pretty cool. That's lysosomes.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and moved through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
All right, I'm going to hit play on this animation. It literally is just showing you what phagocytosis looks like. So you'll see two molecules, or two structures rather. One of them is red, the other is like a fluorescent green. And you should notice that the red ones are being engulfed by the green ones. That is exactly what phagocytosis looks like. Hit play one more time just to run it back. So you'll notice that those red pieces have to be in contact with the green pieces, and then the green pieces are able to kind of like reach out and grab around the red piece and bring them into themselves. So now let's look at everything from the model perspective once again. So here we go with the whole model of the cell. Notice this time the only thing that's colored in are the lysosomes. They're colored in purple. Next to the model, we see a transmission electron microscope visual. They point out where the lysosomes are in the actual slide picture. Underneath of these two, we see up close what's happening. So here's the lysosome here. There's another one here. This outer part right here is the plasma membrane. Recall that I said some kind of molecule, maybe food, a macromolecule, will basically sit on the outside of the plasma membrane of a cell, and then the plasma membrane itself kind of engulfs or takes in the molecule. When it does that, a piece of the cell membrane pinches off and it's completely enclosed and it becomes a vacuole. In this case, they're calling it a food vacuole because it has brought in macromolecules that are made from something that we've eaten. So as soon as it comes inside, notice that the lysosome is like la-di-da on its way. It's probably coming from the Golgi. As soon as it gets on the same track as the food vacuole, the two will combine. So in this state here, so I'm going to do it in black now. In this state, those hydrolytic enzymes, or the proteins that we were talking about, they are not active. Once the lysosome and the food vacuole come into contact, it activates that protein, and so then digestion can occur. You'll notice that the lysosome appears to be completely dissolved during digestion, and really all you need to think about is the fact that both of these things, the food vacuole and the lysosome, the membranes are made of the same pieces. So it's not that the lysosome gets consumed, it's just that the cell membrane that made it up has now morphed in with the food vacuole that came from the plasma membrane. When we talk about a lysosome, we also have to throw in another organelle that pops up. It's called a peroxisome. Let me just say one more thing about lysosomes before I explain what the peroxisome is. Lysosomes are not typically found in plant cells, so we tend to think of them more as belonging to species from the animal kingdom. That's not to say that no plant cells have lysosomes. Some of them certainly do. But the peroxisome is something that we find most often presenting itself in both cell types. So peroxisomes are also used to break down some type of bigger molecule. So in the picture that we're looking at, the transmission electron microscope picture shows you a peroxisome fragment, and then they show you a piece also of a mitochondria. So what happens is these two are going to come together, the peroxisome and the mitochondria. They are enclosed in their own membrane. So they have a vesicle, excuse me, that surrounds them. I'm looking right here. And once the vesicle has everything it needs in it, the lysosome will come. Again, they combine, and then this digestion of this worn out mitochondria happens. So really quickly, let's just talk about peroxisomes with some brevity. I don't want to spend a lot of time on them, but they are worth talking about. So here's what you need to know about peroxisomes. They're membrane-bound vesicles that enclose enzymes that are involved in the breakdown of fatty acids. So unlike the enzymes in a lysosome, the enzymes in a peroxisome have to be synthesized by free ribosomes, and then they can be transported into a peroxisome from the cytoplasm. So when we think about the lysosome pinching off from the Golgi, it is coming with those inactivated hydrolytic enzymes already on the inside. The peroxisome is different. You get the enzyme from something that a ribosome cranks out, and it is engulfed. And so that is just the making of the two are, very different. The other thing is, because the enzyme that is inside a peroxisome is able to what's called oxidize, which means it's able to pull off electrons basically from fatty acids, they produce as a byproduct hydrogen peroxide. So I'm talking about the same type of hydrogen peroxide that comes in that brown bottle if you go to CVS or wherever you're going to buy it. And so what about H2O2? By the way, that's the chemical formula for hydrogen peroxide. What about H2O2 makes it bad? In effect, it is toxic. Like, you can't drink hydrogen peroxide. Like, that's not a thing that you would ever want to do. But peroxisomes, in addition to producing this H2O2, there's a second enzyme that they have present. It's called catalase, and we actually do a lab with catalase and potatoes, interestingly enough, where we look at how catalase is able to break down hydrogen peroxide. So it's like the peroxisomes make this H2O2 that is toxic itself, but they come with a backup to detoxify what they make. So when do you see like this catalase in action? Think about if you've ever cut yourself. If you were to pour hydrogen peroxide onto a wound, then what you should see is a bunch of bubbles like popping up. And what that bubbling is, or that fizzing is actually the catalase breaking down the hydrogen peroxide that you have poured in the cut. So that is my spiel about lysosomes and peroxisomes. We're moving on to the next organelle. They're called vacuoles. So this is what they do. They just store stuff. That is what their job is. You don't find vacuoles in animal cells. You find them in plant cells. Typically, you'll hear them referred to as the central vacuole, or even like a water vacuole. So all it is, it's a compartment within a plant cell. It takes up a lot of space, and the contents inside of a vacuole are never the same as the contents that it bathes in. In other words, the cytosol composition is different from the composition of what is housed inside of the vacuole. This slide gets into the various types of vacuoles that exist. So what's listed: a food vacuole made via phagocytosis. You have contractile vacuoles that essentially bring water in and pump it out as necessary. They help with turgor pressure inside of plant cells. It's what kind of helps the plant cell, or rather helps the plant stem stay straight up. You'll notice that like if you don't water a plant for a couple of days, it'll wilt over. This has a lot to do with that particular, or just the vacuole being full. The other vacuole listed, the central vacuole, so this is the one that I was saying is associated with plants. And then the last bullet points alludes to the fact that there are certain vacuoles that you can find in a plant or in a fungal cell, and they can contain enzymes that are similar to that of a lysosome. So vacuoles could also be for junk or for waste. This figure 4.14, shows you where the plant cell vacuole is located. Again, you have this model cell. They color in the specific organelle that they're talking about, and then in the transmission electron microscope image, they point out where these different organelles are actually located. All right, we're coming to an end on this one, finally, but we did cover a lot of organelles. So anyway, this is just a brief review of everything that we just talked about as far as the endomembrane system goes. This is what you need to remember. You have to remember that when we were looking at all of these various organelles and how they work to complement one another. That's the first thing. The second thing is that we've seen that the endomembrane system, it's a series of membranous organelles that work together, and they communicate by means of transport vesicles. So without these vesicles being able to move things back and forth, there's certain processes that just would not happen, because even though much of it is a continuous membrane, if you will, there are still parts that have to be bridged. So those transport vesicles are going to bridge the gap for us. So here we are at the end, figure 4.15. It's just the review image of the relationship among the organelles of the endomembrane system. So we start out in the nucleus. I want to make this in black. I apologize. We start out in the nucleus. There's spot 1. When you come out of the nucleus from a nuclear pore, you end up in the endoplasmic reticulum. So that's our second stop. The next stop would be maybe a vesicle, so we could call this 3. 4 would be us arriving at the Golgi. Step 5 could again be more of this vesicle idea, moving things out. If we need to break something down, it would happen in step 5 as well. So the very last part of this, so step 6, could be something called exocytosis. In this case, we are making something and we're pushing it out of the cell. We can go the opposite way as well. There could be something on the outside that needs to be brought in, and then a lysosome break it down. That would be called endocytosis. Specifically phagocytosis, when we're talking about breaking down or engulfing macromolecules and then breaking them down. So I hope this was very comprehensive for all of you. In the next one, we will finish more organelles. So happy weekend. Bye.